over 50 million.
Welcome back to the deep dive. Few areas of science have seen quite the explosion that single-cell RNA sequencing, cRNA-seq, has brought. It's let researchers move past just looking at averages from tissue samples. Now they can profile gene expression in individual cells. Painstakingly, yes, but the result, these absolutely monumental data sets, think massive cell atlases like the human cell atlas. We're talking well over 50 million cells already cataloged, but well, that kind of rapid scale creates its own headache, just sheer information overload. And the traditional single cell models, the ones built specifically for this kind of data, they hit two big walls. First, they just don't scale well. Handling 50 million cells and knowing that number is only going up, it's tough. And second, maybe even more critically, they couldn't really integrate all the other knowledge, the research papers, clinical notes, the metadata that gives context. They were basically blind to text. So our mission today is to dig into a really breakthrough framework called C2S scale, that's cell to sentence scale. It tackles this problem head on by using large language models, LLMs, to try and, well, cut through all that complexity and pull out genuinely deep, biologically meaningful insights.
Exactly. The older models, they were just too specialized. They were stuck with the numbers, the expression data. They couldn't speak the language of biology as described in text. Cell-to-sentence, C2S, that's the real innovation here. It finds a way to translate that complex, high-dimensional cell data into a format in text, essentially, that these powerful LLMs can actually understand. It bridges the gap between the genomic data and all that human knowledge written down in papers.
And here's what really grabbed me, the hook that makes this worth diving into. This isn't just theory. It's not just a cool idea on paper. C2S scale has already led to a real validated biological discovery. They used it to find a drug that turns out to work only in a very specific immune environment, a low-level one. And that's a finding that standard lab methods just completely missed. They weren't even asking the question that way.
Mhm.
Okay, let's really unpack this core idea then, because honestly, it sounds a bit like magic. How does a huge LLM, something built for processing language, suddenly learn to read biology from gene expression data? How do you actually turn a cell into a sentence?
Right. Well, it's surprisingly elegant, really. The aha moment was realizing they needed to ditch the raw numbers, at least as the primary input. Instead of feeding the model that huge list of, thousands of genes and their exact expression levels, they transform it. They create what they call a textual cell
A
sentence.
cell sentence. Okay, what does that look like in practice?
So for any single cell, you look at all the genes being expressed, tens of thousands potentially. You just rank them purely based on how active they are.
Hmm.
The highest expressed gene comes first, then the next highest, and so on. And the names of those genes in that ranked order literally form the sentence, like gene, gene, and so forth. The position in the sentence tells the model the gene's relative importance in that specific cell.
Okay, so you're taking continuous data, the actual expression levels, and turning it into a discrete, ordered sequence of gene names. But wait, doesn't that throw away information? Those exact numbers, aren't they important? Feels like you'd lose some precision there.
That's the absolute crucial question, yeah. And they had to show that it works. What they found is that, yes, you lose the exact quantitative value, but the transformation is largely reversible, meaning the relative information, the ranking, captures most of what matters biologically. Minimal practical loss. But here's the payoff, the reason you make that trade. Because now the model isn't seeing numbers, it's seeing gene names. And gene names are words. Words the LLM has already encountered millions, maybe billions of times during its pre-training on, PubMed abstracts, textbooks, all sorts of scientific text. So suddenly, the model can connect the dots. It already knows from reading all that literature that maybe gen X is involved in inflammation and often shows up alongside gene Y. By making the cell data look like a sentence, you unlock all that pre-existing knowledge about biological relationships. It connects what the gene's being expressed with the why from the literature. It's pretty neat.
And they really went for it with C2S scale. This wasn't some small pilot study. The model they built is huge, 27 billion parameters.
Right, a massive model.
Hmm.
And you need a massive data set to train something that big effectively.
Hmm.
They pulled together over 50 million transcriptomes from both humans and mice using big public resources like the Human Cell Atlas. Then they added in all the associated text, the abstracts, the annotations, metadata. It totals up to over 1 billion tokens of training data. Just a colossal amount of information.
And if you look at the results, it ties back into what we're seeing in AI development generally, right? The performance seems to get better just by making things bigger.
Absolutely. That's one of the really compelling findings. They show pretty clearly that as they increase the model size, the number of parameters and the data set size, the performance on key biological tasks just kept getting better. Things like accurately predicting what cell type you're looking at or figuring out which tissue a cell came from. It follows those scaling laws that are well known in general AI and natural language processing, which makes you wonder if they're getting these kinds of results with 27 billion parameters, and we know scaling usually continues to help. What happens when we get to the next generation of even larger models? What discoveries are still waiting?
That scalability definitely seems to translate into versatility, too. The paper emphasizes that C2S scale isn't just a one-trick pony. It can handle prediction, generation, even complex reasoning, all within one system. And importantly, it's not just matching other models. They show it often doing better than specialized models like SCGPT, which only look at expression, and even better than some of the big general-purpose LLMs like GPT-4o when applied to these specific biological tasks.
Yeah, and that's where that synergy between text and expression really pays off. The model isn't just crunching numbers or just processing text. It's doing both. And that allows it to reason about the biology across different levels. And critically, it can explain its reasoning in natural language. That makes the insights much more accessible, right? You don't necessarily need to be a deep computational expert to understand what the model is telling you.
Okay, so give us some examples of that multi-scale reasoning. What does it look like at different levels?
Well, at the most basic level, the single cell, it's great at classification. Like you mentioned, predicting cell types. They reported, was it 95.43% accuracy on immune cell types? Very strong.
Okay.
But then you zoom out a bit to groups of cells, like clusters you might find in an analysis. The model can do something they call cluster captioning. We basically feed it the cell sentences from a whole cluster of similar cells, and it generates a short human-readable description of what that cluster represents biologically. What pathways are active, what the likely function is. It's not just labeling, it's interpreting.
So it's explaining the story of that cell cluster.
Exactly. It's synthesizing the common patterns into a functional narrative,
Uh,
and then you can zoom out even further to the level of an entire data set or study.
right, the data set interpretation.
Yeah. Here it can generate a high-level summary, almost like an abstract for a paper, describing the key findings of a whole cRNA-seq experiment. And what's really impressive is that it generalizes. It can do this effectively even for studies it never saw during training, which shows it's actually reasoning, not just memorizing.
Now, when the model is doing things like generating data, maybe simulating how a cell might respond to a drug, you need a way to evaluate if that generated data is realistic, right? Standard accuracy metrics don't quite cut it. They borrowed an idea from computer vision.
That's right. They adapted a metric called the Fréchet inception distance, or FID, which is commonly used in AI image generation to assess how realistic generated images look. They created a version for single-cell data, scFID. Instead of an image model, STFID uses a pre-trained single-cell foundation model as the judge. It looks at the generated cell data within this biologically relevant latent space and scores how closely it resembles real cells, capturing those complex, higher-order variations. It's a way to measure biological realism for generated data.
Makes sense. Okay, let's talk about space. Understanding how cells are organized, who their neighbors are, that's hugely important in biology. Did they have to build special features into C2S scale just to handle spatial information?
Remarkably, no. They didn't need specific architectural changes. It seems the model learns about spatial organization implicitly, just by seeing the transcriptomes of neighboring cells together during its training. If cells from the same neighborhood are encoded nearby, the model just naturally picks up on those spatial relationships through the language patterns.
Just from the co-occurrence data.
Essentially, yes. And they even pushed this further. They found they could improve its spatial predictions by feeding it external knowledge about gene interactions as text prompts. So they'd give it information from databases like BioGRED, which lists protein interactions, or CellPhoneDB, which maps known communication links between cell types. They just provided this as natural language context, and the model figured out how to integrate that knowledge with the expression data to make better spatial predictions.
Right.
No recoding needed.
That ability to integrate different data types, especially text, leads us right to maybe the most exciting application, predicting how cells respond to perturbations, like drugs or signaling molecules. Being able to do that accurately allows for virtual screening on a massive scale, something that's just not feasible in a wet lab.
Exactly. And that's precisely how they made their big discovery. They set up C2S scale to perform a really clever dual-context virtual screen. The goal was to find immune-modulating drugs that only work under specific conditions, looking for that context-dependent effect.
Okay, so what was the specific context challenge they set up? What were the two conditions?
They focused on the MHCI antigen presentation pathway. That's the machinery cells used to display bits of protein on their surface, making them visible to T cells, a key part of the immune response. They compared how different drugs affected this MHCI pathway in two scenarios. First, an immune context positive setting, where there was a little bit of background interferon or IFN signaling. IFN is a really important immune signal molecule. The second scenario was immune context neutral, basically, no IFN around at all. The idea was that many tissues might have that low level of IFN, kind of a baseline immune readiness, but maybe not enough on its own to trigger a strong response. They were hunting for a drug that could sort of amplify that existing weak signal.
Not start the signal, but boost it if it's already there at a low level.
Precisely. Looking for a helper molecule, not necessarily a primary trigger.
And the virtual screen, sifting through possibilities, which drug popped out as having that specific context split they were looking for?
A kinase inhibitor called silmitasertib, which also goes by CX4945.
Yeah.
It came out as a top hit. The model predicted it would significantly boost MHCI antigen presentation, but only when that low level of background IFN was present. In the context-neutral condition, without IFN, the prediction was minimal effect.
So the model wasn't just saying this drug increases MHCI. It was saying this drug increases MHCI if and only if there's already a little bit of IFN signaling, a conditional effect. That's a much more nuanced prediction.
Exactly. The prediction was that silmitasertib acts as an IFN conditional amplifier, lowering a threshold for response. And this specific conditional role, well, it hadn't really been described before in the literature or picked up by standard drug screens, which usually just test things in one condition, typically context neutral.
Okay, the prediction is fascinating. But the real test is always the wet lab. Did they validate this prediction experimentally?
They did, and they did it rigorously. They took this prediction and tested it in actual human cells in the lab, specifically two different neuroendocrine cell lines, one from Merkel cell carcinoma, one from pulmonary origin. And here's the crucial part for showing generalization. These specific cell types, in these specific conditions, were completely new to the C2S scale model. They were not part of its training data at all.
So it wasn't just recalling something it had seen, it was applying its learned understanding to a totally novel biological scenario. What did the lab experiments show?
The results lined up perfectly with the prediction. When they treated the cells with silmitasertib alone, not much happened to MHCI expression. But when they combined silmitasertib with that low dose of IFN, the dose that barely did anything on its own, they saw a significant amplification, a big boost in MHCI. It confirmed the context-specific hypothesis exactly as the LLM had predicted. A really strong validation of the model's ability to reason about conditional biology. So what does this really mean going forward? I think it powerfully demonstrates that this kind of model, C2S scale, can generate genuinely new, biologically grounded, and importantly, testable hypotheses about context-dependent effects. These are the kinds of subtle biological mechanisms that standard context-blind lab assays are likely to miss entirely. It highlights how adding that layer of contextual intelligence, often derived from text and complex data integration, is key for the next wave of discovery.
This has been a really eye-opening look at how LLMs are moving beyond just, processing language and are starting to fundamentally change how we can investigate biology. So, just to recap the key points for you, the listener. C2S scale is this framework that manages to unify complex genomic data from single cells with the vast world of biological text. It does this by cleverly treating cells as sentences of ranked genes. And this approach shows systematic gains. It gets better as it scales up, just like general LLMs. This gives it this incredible versatility for reasoning about biology across different scales, from single cells up to entire experiments.
And it really feels like just the beginning. The researchers talk about this paving the way towards eventually creating virtual cells, highly realistic simulations we can use for discovery. The C2S framework seems like a foundation. You can imagine integrating even more types of data in the future, maybe epigenomics, proteomics, detailed clinical data, all into one unified model that tries to capture the complexity of biology more holistically.
It certainly makes you think. In a world swimming in data, developing tools that can help us think critically and find patterns is crucial. And the success here really hinges on carefully aligning these powerful LLMs with biological truth, maybe using techniques like reinforcement learning. But if a language model, by synthesizing billions of data points from cells and papers, can predict a drug context-specific behavior better than a standard lab test, well, it poses a fascinating question for all of us interested in these complex systems. What challenging, maybe context-dependent biological puzzle will you think about tackling next with platforms like this?